like? Does anybody have any questions about what we covered on Wednesday about DNA? We are still talking about DNA, but we are talking about how we use DNA in the world of gene expression. And again, gene expression means we take what is in DNA and make a protein out of it, which requires about two things: transcription and translation. And when we express a gene, we only take the gene part and not any other DNA that's around it when we do this particular phenomenon. And just so you can remember, if you don't remember, we go from DNA to RNA, and then RNA to DNA. First step, transcription. And then the second step, translation. If you've never heard this before, does anybody know what transcribe literally means? Can you be a little more specific? Because there's a way to copy something, but the question is, what are you copying into? Is an important thing between translation versus transcription. And so most times when you hear about transcription, maybe you talk about it or think about it with a lawyer. You're in the courtroom. There's a person called a courtroom transcriptionist. What is that person doing? The court reporter? What are you doing right now? If you're writing things down that are coming out of my mouth, you are transcribing. Because you're taking something in one language or one format of the language and turning it into a different format of the same language. From DNA to RNA, it's nucleotide to nucleotide. So we're not changing the language, we are changing the format. Go from DNA to RNA. Translation is going from one language to the next, or a different language. That's something we're all probably familiar with. Let's take a poll. Who hasn't taken their foreign language requirement yet? Oh, good, you guys are. Usually seniors like, I'm leaving until the very end. You're not all seniors, but still. We also appreciate that translation is the process of changing languages. So we go from a nucleotide language in RNA to an amino acid language in proteins. And while we won't dive into all the details of how we do that, we do need to understand a little bit about this process of going from RNA to amino acid in the grand scheme of things. The enzyme that performs the transcription process, there are a couple different things that are needed for transcription, but when it comes to what is actually making the RNA, it's an enzyme we call RNA polymerase. It's not very unique or sexy. It's a pretty straightforward, basic enzyme name. It's a protein or an enzyme that makes RNA. But it is typically a small thing relative to what makes proteins, which is a ribosome, which is a much bigger structure. Just for fun, when people are looking at things under the microscopes, they call everything a body, which is a some. So the ribosome was a structure that was visible with a microscope. A chromosome was a structure that was visible with a microscope. But they called it a chromosome because it was colored when they viewed it using dyes. They call this a ribosome because it contains ribose, which we now know as RNA. Ribosome contains both RNA and proteins. It's a very complex machine. And one of the cool things I think about the way things work on this planet and life in general is a thing that's made of RNA and proteins uses RNA to make proteins. And so it's like chicken and egg. What came first, the ribosome to make proteins from RNAs, or the RNAs and proteins that were used to make the ribosome to make more proteins? If that blows your mind, then it's pretty cool. But what came first? The thing that made the proteins, or the proteins themselves? To understand a little bit about transcription, we need to understand the basic process that goes on. When we talk about genes and transcription, we have to evoke this idea of gene architecture. A gene is a segment of DNA, but that gene is flanked by two other important pieces of DNA called the terminator and the promoter. The terminator and the promoter are only important for the transcription process. And as we see, the promoter is where transcription will begin, the terminator is where transcription will stop. And so the RNA polymerase and some other factors will bind right here to the DNA, or actually right here on the DNA, and go this direction, copying the DNA into RNA. And eventually they'll run into a terminator, which causes the whole thing to stop. So we've got a beginning point and an ending point. Every gene has to have a terminator and a promoter for it to be expressed through the process of transcription and then later through the process of translation. The gene sequence is more important, and we call the gene sequence what's called here in blue. The sequence in black is the complementary piece of DNA. And there is a specific name that is given to the DNA strand that is complementary to the DNA strand that contains the gene sequence. We call this the template strand. Does anybody ever do any sewing or anything that requires a pattern to copy from? That's generally what a pattern is a template. You've got a little picture here. What grade was it? What, first grade, kindergarten, where we make turkeys out of your hand? It's like using hand as a template or something that kind of looks like a bird. Template and the other strands, what we call coding, vary. You can't say it's always the top strand, because top and bottom don't really matter. It's wherever the gene is. The gene is located in the coding strand, and the other piece of DNA is always called the template strand. And why it's called the template strand is when the RNA polymerase is actually making RNA, the RNA that's being made is base paired to the template. Now you don't need to understand all of that. In molecular biology, we talk much more about the actual molecular mechanism of how this works. But as a endpoint for why we care about this process is by using the template strand. The RNA polymerase is going to make an RNA that is going to more or less match the sequence that is the gene sequence. So we make an RNA that is almost identical to the DNA sequence. There's some variations, obviously, because DNA contains T and RNA contains U. But you're basically copying down all of the A's, G's, and C's as they are present from DNA to RNA and change every T to U, and you get your RNA molecule. But the RNA transcript, the thing that's made from transcription, is a direct copy of the DNA sequence. And that's an important theme going forward. Okay. The blue piece and the red piece, the gene and the RNA match. They're not complementary. They are almost identical in sequence to each other. But the template, the black piece of DNA here in this case, is complementary to the RNA, but also complementary to the gene. And you just have to pull apart the DNA to make the RNA. And we're not going to talk about any more mechanisms of how transcription actually works. Luckily for us it does, and it's pretty efficient. RNA polymerase hardly ever makes any mistakes, which is useful. But we make a piece of RNA that is much longer than what we actually need. The thing we only need to make the protein is from the beginning of the gene to the end of the gene. But we have this extra bit on one side, this extra bit on the other. And it turns out that there's a little bit of extra DNA or RNA on both sides that are actually useful. So in every RNA that's produced, we divide it into 3 basic regions. The most important of which is in the middle, which we call the ORF, the open reading frame. We define an open reading frame as there's a start codon and a stop codon some distance away from each other in the right frame, meaning sets of 3s away. We don't need to understand all this just yet. But that's what open reading frame is, or it's basically it's the gene sequence in RNA form. We call it an ORF rather than a gene. So gene is a DNA term. ORF, sometimes you use it with DNA, sometimes you use it with RNA. It's kind of an interchangeable thing. When we talk about DNA and RNA, we use phrases like upstream and downstream to denote relative position. Has anybody ever been on a float trip down the river? I mean, downriver is the same reason, because what you go from one part and you travel down. The flow of the water pulls you from one place to the other. If you want to go against the natural flow of water, you have to spend a lot more energy to go in the opposite direction. So as you're standing on like a river, if you're looking upstream, are you looking at the start of the river or the end of the river? Right? That's what we mean by upstream. We're looking sort of where's the beginning. When we're looking downstream, we're looking where's the end. So you just use, if we're at a certain spot in DNA, we look upstream, these are going towards the 5' side. If we look downstream, we're going towards the 3' side. And we call the sort of places that aren't the ORF the 5' UTR or the 3' UTR, meaning untranslated region. So we transcribe more DNA than we actually translate, which actually there's a lot more DNA that's transcribed than is actually translated. We'll get more into that as we go forward. But the 5' UTR and 3' UTR do have some important information. They're not used to make the actual amino acid sequence of a protein, but they do tell the ribosome where to start and where to end. Just like promoter and the terminator tell the RNA polymerase where to start and where to end. That's all you really need to remember about the 5' end and the 3' end is that's the beginning for translation somewhere in there, and somewhere in here is the end of the translation process. For gene expression, the transcript that's made is called the messenger RNA. We'll talk more about how translation actually works in a little bit. But we also use terms like primary transcript and mature RNA to denote some differences between a eukaryotic process and a prokaryotic process. But the RNA that is made directly from transcription without doing anything to it is always called the primary transcript, because it's the RNA that is first made from this process, or it's the most important RNA that's made. But even that's a misnomer. But why I bring it up is in prokaryotic organisms, the RNA that is produced directly from transcription is the primary messenger RNA, and it is used directly and immediately for translation. There's no type of processing or modification of the RNA that's needed. You make it, you use it, you get proteins produced much quicker. Bacteria and archaea are very quick at making proteins, much more quicker. It's not really correct English, but bacteria make proteins a lot faster than eukaryotic organisms like us could ever do. A gene is a DNA term. When we talk about a transcript and how many ORFs are present in a transcript, we use the word cistron. So cistron refers to the RNA. Gene is DNA, cistron is RNA. And I'll repeat it a few more times because some of you are going to either forget it or not remember it correctly and get it wrong on a quiz or an exam. But RNA is the cistron. And I forget where the word cistron came from, but this is where we are. Cistron and RNA go together. Monocistronic versus polycistronic refers to how many open reading frames are contained within the primary transcript. And as we see in bacteria, there are some RNAs or some transcripts that contain multiple open reading frames. And there's a particular name for the DNA structure that allows this phenomenon to occur. But bacteria and archaea will be able to produce two different types of RNAs, monocistronic forms and polycistronic forms, whereas in eukaryotic organisms, we only ever see monocistronic RNAs. Each RNA only contains the information for one protein and not multiple proteins. And part of the reason for this is eukaryotic organisms don't use the primary transcript. They have to do a lot of modifications to the RNA before they can actually use it. And we'll go over what those modifications are in a little detail as we go forward in these slides. We first want to just look at this idea of polycistronic. And how do we get an RNA that contains multiple open reading frames, or an RNA that can produce several different proteins at once? And the basic idea is we get a DNA architecture called an operon. So operon is DNA, polycistron is RNA. It should be very easy, hopefully, keep them straight, operon DNA. And an operon is a chunk of DNA with multiple genes controlled by a single promoter and a single terminator. There's a picture on the next slide we'll look at in a second. But we can organize our DNA. I won't draw a picture because I got a picture in a second. But we can organize our DNA where every gene has its own promoter or terminator, in which case those would make monocistronic RNAs. And in this piece of DNA, we get two monocistronic RNAs. One for gene 2, one for gene 1. Each one would be used to make its own protein through the translational process. But bacteria and archaea will group together genes that are needed for a common purpose. And to make sure they all get expressed at the same time, and typically the same degree or rate, they're grouped together in the DNA. So here you have 4 genes, all controlled by a single promoter. So RNA polymerase binds here, transcribes all 4 of these open reading frames or genes into the RNA, making 4 open reading frames, which can all be translated independently of each other from the same piece of RNA, which is pretty neat. Bacteria are pretty cool. You should not study humans, you should study bacteria. Bacteria are the best. As a person that studies bacteria, I endorse this message. So the operon, as I mentioned before, has multiple genes. Sometimes there's only 2 genes in an operon. There are some that have more than 12 genes in them. So they're really long operons sometimes. But typically, the genes that are in that operon are for the same thing, like individual steps of a biosynthetic pathway, like making amino acids, takes several different enzymatic reactions to happen. Put them all together so they all get made at the same time. Or maybe there are things that, there's some enzymes that have a quaternary structure. It means they have multiple different parts that go together to form a bigger thing. And it's good to have those in an operon because you make both at the same time. And we have multi-subunit enzymes in eukaryotic organisms. We have multi-step pathways in eukaryotic organisms. But for whatever reason, life has evolved with eukaryotic organisms being inefficient, I would call it. Because there's an easy way to do it. Bacteria do it, they do it really quickly. But the eukaryotic way is the long and complex and convoluted way of doing things. Which is why we actually study bacteria to try to understand how things happen, and then go study eukaryotic organisms to see if they're different and how different they are.
So eukaryotes don't have operon structures?
No. And as we'll see in a second, the RNA is chopped up and glued back together, and things are added to the beginning and the end for it to be used for translation. So you can't do operons because you need each one sort of can do something to it anyways. I don't know, this is weird. But again, operon is DNA. An operon will make a polycistronic RNA. A lot of people just get these 2 things confused. They think the operon, they call that the polycistronic RNA. But anyway, you talk about DNA, the DNA organization that contains multiple genes and will allow us to make an RNA with multiple ORFs. The DNA is the operon structure, the product is the polycistronic RNA. We're going to skip, or actually we're going to put an interlude of information in between talking about what eukaryotic organisms do with their DNA. I forget exactly why I threw this in here now. There's got to be a reason. I just can't think about it right now. A gene is a small segment of a chromosome. A chromosome is a large piece of DNA. And again, the chromosome is going to contain many genes. Most of them are monocistronic, but a few of them are going to be polycistronic. It's not really important for anything that we talk about in this class exactly, but the chromosome structures are different in eukaryotic organisms and prokaryotic organisms. They're linear in eukaryotic organisms and circular in prokaryotic organisms. Also, we see some pretty big differences in size. Chromosomes in prokaryotic organisms are always smaller compared to the size of the chromosome in a eukaryotic organism. When you make a linear chromosome, there is a problem that's going to happen because of the linear structure of DNA. And what has been evolved over time to protect the DNA of the chromosome is to put things at the end. These things are called telomeres. The telomere structure is pretty cool. We're going to talk about it as we go forward. I'm going to say that a lot. We're going to talk about this as we go forward. Until we get to it, then I'll stop saying, well, we'll talk about this more. But telomeres are pretty neat structures. And we don't have telomeres in a circular piece of DNA because it just goes around and connects. But on a linear piece of DNA, you have these things at the end that are going to serve as a purpose on the end. We also don't talk a whole lot about prokaryotic chromosomes, and they don't have centromeres in the same sense that a eukaryotic chromosome has a centromere. But the purpose of the centromere is the place where we can attach machinery used to move the DNA around during cell division. There is a similar type of phenomenon that goes on with bacteria. We just don't call it a centromere in the DNA that we do with eukaryotic organisms. And that means when we talk about prokaryotic chromosomes, there's a lot more terminology to describe the different parts of the chromosome than when we talk about a prokaryotic piece of DNA. An operon allows the cell to save space, because you can have multiple genes controlled by one promoter and one terminator, which means you eliminate extra promoters and extra terminators needed, which saves DNA space. That's not what this slide is about, but it's about similar to what this slide is about, which is why the operon structure leads directly into this slide now. Operons allow bacteria and archaea to save space. And there's a reason that this is useful because of this idea of gene density. How many genes does an organism have for a unit length of DNA? Basically, how good are you at using your storage space? Is what gene density is kind of like. If you're really good, you can pack a lot of things into a small little area. If you're really bad, you get one or two things in a really big suitcase. So what we see is as we go from one type of organism to another, we see that the ability to use our DNA efficiently goes down, meaning gene density changes quite a bit. And the prokaryotic organisms have by far the highest gene density, meaning they have the smallest amount of unused DNA. When we talk about gene density, we see how much of the chromosome is devoted to making protein or functional RNA material. And you see, most of these are eukaryotic organisms, but as you get more and more complex, you get less and less good at using the DNA that you have available to you, or you get more and more extra stuff in your DNA that doesn't actually turn into protein that we need to think about. And just for illustration here, you can see the gene density differences between a bacterium and a typical eukaryotic like us. 1,000 pieces of nucleotides versus 100,000 nucleotide difference. It's a pretty big difference. Does anybody have any idea how long a gene, an average gene, would be as far as DNA length? What do you think? I'll give you a hint. It takes at least 3 nucleotides to make 1 amino acid. And the average size of a protein is usually around 300 amino acids. We can do that math in our head really quickly. It's actually 903. It's a trick question. As we'll talk a little about later, you'll hear more about in molecular biology. The last 3 nucleotides is a stop codon, which doesn't actually make an amino acid, but it still needs to be there. So actually, amino acids times 3 plus 3. I don't really care. But generally speaking, we call a kilobase, a KB, is 1,000 nucleotides. It's about 1 gene. So there's 1 gene in 100 times the space. So what else is there? For the longest time, it was just called junk. But it turns out that some of that junk is actually useful. That'd be like American Pickers, the TV show on HG Channel. Someone's junk is someone's treasure. Who's ever driven around and said, oh, there's a nice piece of furniture sitting on the side of the road that they're throwing out? Hey, you know, sometimes good stuff is found in weird places. So when you think about gene density, there is again kind of a link between the operon structure helps bacteria be more efficient, so they have higher gene density. It's sort of they lose less of their DNA, can make more proteins for a smaller amount of space. A lot more monocistronic RNAs and genes, basically all of them monocistronic in eukaryotic organisms. So lots of promoters, lots of terminators, lots of pieces of DNA used up for non-protein stuff. But also, every gene in eukaryotic organisms has 2 types of information. Good information that's used to make proteins, and not so good information that is not used to make proteins. We call the good parts exons. So every gene contains an exon part and an intron part. And it's usually several exon parts and several intron parts. And they alternate. If you sort of think about what a gene might look like in eukaryotic organisms. You have good parts, and then you have these bad parts. It's not really bad, but you've got stuff you need to make a protein, and stuff you don't need to make a protein. And the blue parts would be the exon parts, the black parts here would be the intron parts. But the presence of introns makes all of the genes significantly longer than we would find in a prokaryotic organism. What you might want to ask yourself is, why? Sometimes the question you don't have an answer for, but why? If it's good enough for bacteria, why isn't it not good enough for eukaryotic organisms?
We must need the introns for some reason.
It must have some purpose, or did have some purpose at some time or another. Maybe it's just large cell size needs this other stuff for whatever reason. It's hard to say, but if we look at Carl Woese's phylogenetic tree, bacteria prokaryotic, no introns, archaea prokaryotic, no introns, and then they split. Archaea go this way, eukaryote go this way. Introns happen here, but not over here. And so something happened, evolutionarily speaking, to make introns a thing. I don't know if we'll ever figure it out, but we do have some kind of stories that may be the origins of introns. Maybe I'll tell you. We'll see. When we talk about introns and exons, there is one important idea to think about. Is this a transcribed piece of DNA, and is this a translated piece of RNA? Introns and exons are both present in the DNA molecule. So they're both going to be transcribed. But only one of them will be translated, and that is the exon sequences. Which means any introns that are present have to be cut out of the RNA, and the exons have to be glued back together. We call this the splicing process. It's a pretty cool mechanistic reason or mechanism of how the RNA basically cuts itself. Actually, it doesn't cut itself, there's RNAs that cut it for it. But still, it's a fun story. Imagine you have to make this really long thing and then cut parts out. and then glue the parts together correctly, in the right order. Because you put them in the wrong order, you make the wrong protein. Or you make a different protein, which is sometimes useful. But we keep the exons, say the exons are both translated and transcribed, but the introns are thrown away so they only get transcribed but never translated. And so we go from what's called a primary transcript that contains introns to a mature RNA that will only contain exon sequences. There are two other things that are done to make the RNA a mature RNA, and that's to add a cap to the front and a tail to the end. Later on in this semester, and I'll just say later on this semester, I've done it quite a lot, the tail will be important as a regulatory mechanism to control the RNA and how long it lasts. The cap is just used for translation. It just sort of tells, here's where you start translating, so it does have some purpose. But we don't need caps and tails in bacteria, so obviously you can get away without it. So why the extra complexity? I don't have an answer. Your guess is probably as good as mine. But it is something you need to keep in mind, that overall eukaryotic organisms are just much more complex than prokaryotic organisms. When we compare one organism to another, we do see that there are differences in the numbers and lengths of the introns that are typically present. And so in what we call simple eukaryotic organisms, we have shorter introns and there's typically fewer of them. But as we get into things like humans, the introns get much longer and we get to have a lot more of them. And so longer introns means shorter exons sometimes, and also many more exons to glue together. But again, why? If it's good enough for simple eukaryotes, why isn't it good enough for us, complex, higher order eukaryotic organisms? I have no answer. I just have questions. Again, because of the ability to make operons in prokaryotic organisms, we can have both polycistronic primary transcripts and monocistronic primary transcripts, which are used directly and immediately for translation. For eukaryotic organisms, all of the genes have their own promoters and terminators, so they're all monocistronic. And there's significant processing that has to happen before the RNA can be translated. The other thing that's not listed here that's also somewhat important is the fact that in a eukaryotic organisms, the DNA is inside a nucleus, which is a membrane enclosed structure. And all the ribosomes are outside the nucleus. So any RNA you make inside the nucleus must travel or be transported to another location inside the cell before you can actually translate it. It's not needed in bacteria, no nucleus, also a really small cell, so things move around much quicker. And so bottom line, again, I mentioned this earlier, bacteria able to make proteins much faster than a eukaryotic cell. And as a byproduct of this fast protein production, prokaryotic cells divide much quicker, on a faster pace than a eukaryotic cell could ever do. There's one more aspect to the idea of gene expression that we need to think about, and that is the direct relationship between the DNA sequence and the protein sequence. This is a phenomenon that we call collinearity. We can point to the DNA at a position and say that corresponds to a certain position in the protein. And if collinearity exists, point A is next to point B, and C and D and E, in both the DNA sequence and in the protein sequence. And that's always true for prokaryotic organisms. We call it universal. That's what it's going to say in the next slide. Collinearity is always universal in prokaryotic organisms because we go directly from DNA to RNA to protein. Nothing is removed, nothing is added, nothing changes. The nucleotides are always next to each other, which means the amino acids are always next to each other. But when it comes to eukaryotic organisms, collinearity is not considered universal. It will have pockets of what we call universality, I guess is the best way to say it. That's over an exon. Between beginning of an exon and the end of an exon, there would be collinearity between the DNA and the protein. Amino acid 1 is next to amino acid 2. Codon 1 is next to codon 2. But at some point or another, an intron is going to interrupt amino acid 20 and amino acid 21. So they won't be next to each other on the DNA sequence, but they end up next to each other in the protein sequence. So collinearity has been broken because there's a part of the DNA that was taken out before the protein was actually made. It makes it a little bit harder to sort of find things in eukaryotic organisms because there's the bits that are interrupting our gene sequences. But for bacteria, it's pretty easy. You basically can reverse engineer a protein into a DNA sequence and try to find that DNA sequence in a genome. And you can't really do it with the proteins of eukaryotic organisms because you don't have the introns to sort of fill in the missing pieces. So just as a rule, it's just a basic piece of fact. This collinearity, present one, not present in the other when we think about things. But it's all sort of tied together. Introns and exons sort of break up the normal way in which we do things. I think I left these slides in your set of slides. I don't remember if I did or not. Okay, good. There's probably a reason for that. Usually so I could talk about it and you can see it, because some of this is fine information. Back when the Human Genome Project started, you want to guess how long they thought it might take to actually sequence an entire human being?
4 years.
I would say out of 0. Might be closer. They really thought it would take 10 or 20 years to do it. Basically because the sequencing technology wasn't that great and the speed of it wasn't that great. The technology, as you might notice with phones, goes really quickly. So once the first sequencing machine is made, they got a lot faster and a lot better, and it took maybe 5 years to really do most of it. But there were a few little parts they had to sort of finish. When you sequence a eukaryotic organism, there is a fundamental problem that has to be solved. You have 2 copies of every chromosome. So how do you put together a puzzle that's actually 2 different puzzles? And some pieces may actually, could be in both puzzles, but really only one puzzle, not the other puzzle. Try it next time. Buy 2 puzzles that have a similar picture, and throw all the pieces together in one box. And then try to make your 2 puzzles together. So when we first sequenced the genome of some individual, we got most of it, but we didn't get all of it. And what we did get is a type of DNA that we call euchromatin, or the true chromatin. But to get to the rest of every bit of chromosome, it took 7 more years. And basically, I think we're done. They said it was done in March 2022, where we sequenced every single nucleotide in someone's genome. And again, I haven't talked about genome, but genome means all of your DNA, not just a single chromosome, but all of your chromosomes. And so for humans, that means all 46 of your chromosomes have been sequenced from end to end. And they call this the telomere to telomere group, sort of to get from one end to the other. And why are telomeres problematic? Because that's what we didn't have before. Because they are heterochromatin. And the centromeres are also a heterochromatin, which is a lot of repeat DNA, which we'll hear much more about as we go forward in a little bit. But also what helped them finally finish it, they found they used a haploid cell rather than a diploid cell as the origin of the DNA. So now you only have 1 copy of your chromosomes, it's a little bit easier to sort of put things together. And what a lot of people want to do is sequence as many people as possible and combine all of those genomes together, try to figure out what is sort of a wild type of a human. And if you compare a lot of individuals, you can see there are differences, and we'll talk about what some of these words mean as we go forward, probably all of them. You can see if we compare a number of different individuals, you see there's some variation in one individual from another. And so sequencing one person is not enough, because there's a lot of variety in humans. We can sequence enough individuals from every walk of life, every region of the planet. We need to get a whole lot more sequencing, really figure things out. Now, getting back to the idea of gene density, what is taking up the rest of the space? Introns take up some of the space, so that lowers our gene density ability. There's a whole lot more extra space out there that we need to account for. And it turns out that most of that other space is transposon DNA sequences. And transposons are these really small pieces of DNA that can move themselves. They can cut themselves out of DNA and cut themselves into another location. They pop out and into somewhere else. And just like in the case of the P we looked at earlier, if you put a transposon into a gene, that gene basically becomes non-functional. So if these things move in the wrong place or the wrong time, it can cause a lot of problems. The first scientist that identified these is Barbara McClintock. She was studying corn, or actually maize, which is the wild form of corn. And the reason why she identified them, or the question she really the question that she really wanted to answer is, why are there blue kernels and red kernels and white kernels and yellow kernels in the same ear of corn? So she analyzed the DNA and sequenced them and found out that in some of them there were transposons in some genes that made the kernel blue or red or white or yellow. It turns out these are universal. Humans have these things as well, and we have a lot of them. Luckily for us, I guess I would say all of them, but I might be wrong, are non-functional, meaning they don't work anymore. They did at some point in the past. And some of the evolutionary studies that have been done to try to analyze human genomes and the genomes of other animals suggest that it's like more than 1 million years ago that transposons stopped working in the eukaryotic lineage. So they were there for a time, and maybe they're jumping around, was all the phenotype differences that we saw and get. But estimates are that almost half of our DNA is transposon DNA. Think about that. You're only 55% human. Which isn't even true, because you're only 1% human to start with. Because about 90% of you is bacterial cells. So you're 0.5% actual human, based on DNA sequence. A transposon transposes through the process of transposition. Those are the terms we use. A transposon will transpose, meaning it's moving. And it moves through the process we call transposition, because it moves from one location to a new location. I think we're almost out of time, so we'll have to pick up this slide on Monday to talk about the 4 different types of transposons that we find in the human genome. It'll only be this slide that'll go on to talk about what these things look like and how they might be